Hello, today Nanagarn. Today we're going to be solving October-November 2023 Paper 1 Variant 2. And this is going to be Part 2. Don't forget to check out Part 1. And before we start, I'd highly appreciate it if you would take a moment to subscribe to this channel and like this video. Also, don't forget to check out our website in the description box down below, which you can download your free topic 1, 2, 3, and 4 notes. All you have to do is log in into an account and press File Share to download your notes. Question number 21. Which statement correctly describes a feature of a DNA molecule? So we all know that DNA molecule is a polynucleotide. These are two polynucleotides anti-parallel to each other. And each nucleotide is made from a nitrogenous base, a pentose sugar, and a phosphate group. Now let's see the suggestions that we have here. A, it has two parallel strands twisted into a double helix. So parallel is incorrect, they are actually anti-parallel. B, it has complementary bases all held together by 3 hydrogen bonds. Now this is incorrect. We all know that we have adenine and thymine, then cytosine and guanine. So adenine and thymine are held by 2 hydrogen bonds, while cytosine and guanine are by 3 hydrogen bonds. Therefore, this is incorrect. C, it has phosphodiester bonds that are formed using DNA ligase. So a phosphodiester bond is a bond between the pentose and the phosphate group and the pentose sugar, and it is formed directly by the DNA ligase. Therefore, this is correct, and it's a very strong covalent bond, and the DNA ligase forms the backbone of the DNA molecule. Now for D, it has a backbone of phosphate molecules and nucleotides. So this is incorrect because it has a backbone of the phosphodiester bond is what forms the backbone. Therefore, this is incorrect. And the answer is going to be C. And it's good to mention that the backbone is formed by the sugar and the phosphate, not the nucleotides. Therefore, this is incorrect. And the answer is going to be C. Number 22. Which diagram correctly shows the replication of DNA? So, we all know that DNA replication takes place by the enzyme DNA polymerase. And as we all know, we have 2 types of strands. First of all, it's the leading strand and the lagging strand. We all know that DNA polymerase replicates from the 3' to 5' direction. And this is the template for the leading strand. So from 3' to 5'. And for the lagging strand, it's the opposite. So the template, so the actual molecule that's going to be replicated must run from the 5' to the 3' direction. Therefore, the DNA molecule that's going to be formed is going to run from the 5' to 3', and the lagging strand is going to run from 3' to the 5'. Now, there's something worth to mention here. You know that DNA polymerase only adds nucleotides from the 3' to the 5' direction, which makes the leading strand. This means that for the lagging strand, if it were to start from here, it has to keep going back and forth, This leads to something called Okazaki fragments, because DNA polymerase keeps going back and forth. Therefore, these are joined by DNA ligase enzyme. Now, since we mentioned this point, let's see the suggestions that we have here. As I just mentioned, do not get confused in a point. Now, I know that the leading strand is from the 3', so the template would always be from 3' to 5', and the lagging strand, the template would be from 5' to 3'. This is only regarding the DNA polymerase, how it replicates. But the direction of replication must always be from the 3' to the 5'. Always, because as I said, for the lagging strand, DNA polymerase keeps going back and forth. So because it starts at the template from 5' to 3', it keeps going back and forth. back and forth. So the direction of replication must always be from 3' to 5'. Therefore, let's see the suggestions that we have here. We're looking for the leading strand and the lagging strand, both from 3' to 5' and 3' to 5'. Therefore, the answer is going to be C. The reason for this is that DNA polymerase itself must always replicate in that direction, and regardless, even if the lagging strand starts at template 5' to 3', it must always go back and forth to replicate in the same direction. And this is what causes the Okazaki fragments and causes the enzyme DNA ligase to stick them back together. Question number 23. The RNA codon wheel is a tool used to find which amino acid will be translated from mRNA sequence. Codon position 1 is in the center of the wheel. Codon position 2 is in the middle of the wheel. And codon position 3 is near the edge of the wheel. So here. The 3 letters on the outside of the edge of the wheel identify the amino acid. The diagram shows a section of DNA coding for one amino acid. The template strand is outlined in a box and the DNA base sequence is read from the 5' to 3' direction. So this question actually looks harder than what it is, so let's break it down. First of all, here we could see the template strand, so thankfully they've identified it for us. So as we all know, if we can see the phosphate group here, this means that it starts at the 5'. And the 3' is going to be the pentose sugar. So we know that it goes from the 5' to the 3'. Now, let's zoom in a little bit. We have purines and pyrimidines. The purines, we have adenine and guanine. For pyrimidines, which are a lot smaller, we have thymine and cytosine. Okay, so purines have 2 nitrogenous bases, whilst pyrimidines is only 1. And they're a lot smaller. So let's go into the next point. So that's the first point we have here. Regarding the hydrogen bonds, we know that adenine binds with thymine in 2 hydrogen bonds, and cytosine binds guanine in 3 hydrogen bonds. Now this makes it easy to identify what these bases, nitrogenous bases, could possibly be. So first here we could see 2 hydrogen bonds. This means it's instantly either an adenine or thymine, because here we could see 2, therefore it's a large molecule, therefore it must be a purine. A purine, which has 2 hydrogen bonds, must be the adenine. And here it's either cytosine or guanine. Here it's the pyrimidine, therefore it must be the cytosine because it's smaller. Same with here. Now this is a DNA base sequence. Now we have to translate this to mRNA because this amino acid wheel is for RNA codons. So if you were to translate this to RNA, so it's going to be uracil. It's going to turn into uracil, guanine, and guanine. That's all we need here. And we know that this is going to be the first codon because it translates from the 5' direction. And this is going to be the third codon because mRNA molecule is translated on the ribosome from the 5' to the 3' direction. Be careful for this point. Therefore, we want to look for this codon, UGG. Now, let's take a closer look here. It's going to be U here, G, G. Therefore, it's going to be tryptophan. And the answer is going to be C. Question number 24. 4 students sketch diagrams of the apoplast pathway and the symplast pathway. Which sketch is the most accurate diagram of the 2 pathways? So first of all, regarding the apoplastic pathway, it's the movement of water outside the cell through these cell walls. So the water doesn't actually enter the cell, and it's a passive process. However, the apoplastic pathway is blocked by the Casparian strip, forcing all water to move by the symplastic pathway, which actually requires more resistance and it's way slower than the apoplastic pathway, which mainly consists of moving outside through the cell surface membrane. And now we have the symplastic pathway, which the water moves actually inside the cell by osmosis through the vacuoles. And this pathway is an active and it has more resistance. And water goes down water potential gradient. Now let's see the suggestions that we have here. So the symplastic pathway does not go through the cell wall, therefore this is completely incorrect. Same with here, this is incorrect. Now let's look at these two. Here the apoplastic pathway goes outside the cell wall, this is correct. However, here this is incorrect because it assumes that the apoplastic pathway is still maintained by the Casparian strip. However, the Casparian strip blocks the apoplastic pathway and forces the water into the symplastic pathway. Therefore, this is incorrect. Now, for finally D, here we could see the apoplastic pathway through the cell walls and the symplastic pathway through the actual cell, and then the Casparian strip blocks the apoplastic pathway. Therefore, this is correct, and the answer is going to be D. The photo micrograph shows some xerophytic adaptations in leaf tissue, which shows the correct functions of structures 1, 2, and 3. So first of all, let's start by identifying these structures. The first one is called the trichome. The second one is a curled leaves or rolled leaves. And 3 is a thick, waxy cuticle. Now, the trichome and curl leaves have very similar functions. They create a layer of still air, which traps humidity, therefore reducing the water potential gradient and trapping air, creating a layer of humidity. And this reduces water vapor evaporation. So, let's start by exclusion. First of all, we know that 1 and 2 traps a layer of moist air, so this is correct. Thick waxy cuticle does not trap a layer of moist air, it only reduces evaporation. Now, it reduces evaporation from the epidermis of the leaf, technically all 3 of them, because the trichome and curl leaves creates a layer of still air, which creates humidity, therefore reduces the water potential gradient and reduces evaporation. A thick waxy cuticle creates a barrier, therefore also reduces evaporation, therefore all 3 of them must be correct. Which leaves us to the last suggestion, therefore the answer is going to be B. Question number 26, which processes are required for the transfer of assimilates from a sucrose cell into a phloem sieve tube? So let's quickly outline the process. So this is a companion cell, and this is the sieve tube element. Now, outside the companion cells are found these sucrose molecules. Now, what the companion cell does is that they have pumps which pump hydrogen, so carrier proteins which pump hydrogen ions outside to its cell wall. This is by active transport, or an active process in general. Now, the sucrose molecule combines with a hydrogen ion, so not technically combined, they just go back inside the companion cell together along something called a carrier protein by facilitated diffusion. By facilitated diffusion. Now, once the sucrose is inside the companion cell and there's a higher concentration of sucrose inside the companion cell, it goes by passive diffusion into the sieve tube element down concentration gradient through the plasmodesmata. So there should be plasmodesmata here. It goes by passive diffusion down concentration gradient, Therefore, all 3 of these processes are involved in the assimilate transportation through the sieve tube element. Therefore, the answer must be A. Question number 27. When sucrose is loaded into the phloem, it has to travel from mesophyll cells to a companion cell and then into the phloem. Which row shows the relative concentration of sucrose in each type of cell in order for this process to take place? So, going back to the last question and our last point, here we have the phloem sieve tube element, the companion cell, and the mesophyll cells, which contain the sucrose, and they are outside. Now, as I mentioned earlier, that the companion cell pumps hydrogen ions to its cell wall and then combines with sucrose, not really combined, but they go back in together. So hydrogen down its concentration gradient and sucrose against concentration gradients. Now this active process, going back in through a carrier protein, creates a higher concentration of sucrose inside the companion cells. So companion cells must always have the highest concentration of sucrose due to the active process of pumping out hydrogen ions and then the passive process of these hydrogen ions going back in with a sucrose inside the companion cells by facilitated diffusion. Therefore, the sucrose goes against concentration gradient. Therefore, sucrose accumulates into the companion cells. Now, because companion cells now must always have the highest concentration of sucrose, we all know that sucrose now goes down concentration gradient by passive diffusion, by normal diffusion, into the phloem sieve tube elements, down concentration gradient. Therefore, the phloem sieve tube elements is going to have the second highest concentration of sucrose. And now the last concentration, or the lowest, is going to be the mesophyll cells. Now here we're looking for the row which has the highest concentration of sucrose into the companion cells compared to the other phloem sieve tube elements and mesophyll cells. Therefore, this is the only logical measurement. Therefore, the answer is going to be B. Question number 28. Which feature of transport in plants is correct for xylem and phloem? So both. So let's see the suggestions that we have here. A, it is passive. This is incorrect because transport in the phloem is partially an active process in the companion cells, which includes actively pumping hydrogen ions outside into its cell wall. Therefore, both of them are not passive. Therefore, this is incorrect. B, it occurs by mass flow. Yes, this is correct. It occurs, both of them, by mass flow with large quantities going in one direction. Therefore, this is correct. One direction for the xylem and both opposite directions for the phloem. It occurs by mass flow. C, it occurs from source to sink. Now, this is incorrect because the source of the xylem is the roots and the source for the phloem is the leaves and mesophyll cells. Therefore, the source and the sink is incorrect. They're not always the same. And sometimes they swap, especially in the phloem. Therefore, this is incorrect. D, it occurs in only one direction. Now, this is also incorrect because the phloem is variable. It goes in both directions. It's a bidirectional transport. However, xylem is what goes in one direction. Therefore, this is incorrect, and the answer is going to be B. Question number 29. The diagram shows an anterior view of a vertical section through the human heart. Which row shows the correct labels for the diagram? So, let's quickly start labeling. So, first of all, you must identify where's the right and where's the left. We know that the left side of the heart is more muscular. So, we could see a more muscular wall here. Therefore, this is going to be the left side. The reason for this is that the left ventricle has to generate a larger pressure and a larger force in order to push blood to the entire body, in contrast to the right side, which has to push it to the pulmonary system. Therefore, now we know our left, which is this side, and our right. Now, let's start by labeling number 1. We know that it goes to the pulmonary artery. So, the right ventricle pumps blood into the pulmonary artery, which is deoxygenated blood, so 1 must be the pulmonary artery. And for 2, we know that all the valves, whether it's the aorta or the pulmonary artery, the valves leading from the ventricle outside is always going to be a semilunar valve. So these are semilunar valves. And now for 3, we know that the valves between the atrium and the ventricles are genuinely called atrioventricular valves. But for the right is tricuspid and for the left is bicuspid. So 3 is going to be atrioventricular valves. And finally, for 4, we know is going to be the vena cava. The reason for this is that the vena cava returns the blood into the right side, into the right atrium. Therefore, let's see the suggestions that we have here. We have pulmonary artery, the semilunar valve, then atrioventricular valve, and then finally the vena cava. Therefore, the answer is going to be C. Question number 30, what does the inner layer of veins and capillaries contain? So, we know that veins contain 3 layers, which is tunica intima, tunica media, and tunica externa. The tunica intima have an endothelium layer, and then the tunica media has variable elastic fibers, muscle fibers, and collagen, and then the outer layer has mostly collagen. This is only regarding veins and arteries. However, capillaries only contain 1 layer, and it's going to be the endothelium. Therefore, here it's asking about the inner layer of veins and capillaries. The inner layer must always be the endothelium, and it is the tunica intima in the veins and the only layer in the capillaries. Therefore, endothelium is correct, and the answer is going to be C. Question number 31, the graph shows the changes in blood pressure and water potential across a blood capillary from arterial end of the capillary to the venous end of the capillary. What could result in an accumulation of excess fluid? Okay, a rule of thumb that we must always know is as blood goes from the arteries to the veins, or as blood leaves the heart back to go to the veins, the pressure keeps decreasing and decreasing and decreasing. Now, what could result in an accumulation of excess fluid? Now, let's see the suggestions that we have here. A, an increase in concentration of large proteins in blood plasma. Now, this is incorrect. So let's think scientifically. Here we have 2 forces. So in an arteriole and then going to capillaries, we have 2 different types of forces. First of all, we have the force of the blood itself and the pressure, and then we have the proteins. What I'm trying to say is if you have an increase in the concentration of large proteins in the blood plasma, guess what's going to happen? Then water is going to go down water potential gradient. Therefore, this means that water is going to leave the tissue fluid and go back into the capillary. The reason for this is that the water potential gradient at outside is greater. Then it's going to go back in because there's an excess of large protein down water potential gradient. Therefore, this is incorrect. B, an increase in blood pressure. This is correct. The reason for this is I said there's 2 types of pressures, the hydrostatic pressure and the pressure of the proteins in general. So as you increase the pressure, imagine that we have capillary pores. And the moment you increase the force of pressure, you're pushing more against these capillary walls. Therefore, this is going to force more fluid outside. Therefore, this might be correct. Let's see the next. C, a decrease in the concentration of small proteins in tissue fluid. So this is also incorrect. Let's assume that we decreased the amount of proteins. So we didn't increase the concentration inside the blood plasma. We decreased the concentration of small proteins into tissue fluid. This means that now outside there is a higher water potential gradient. This means that water is going to go back into the capillary down water potential gradient. Therefore, this would not cause accumulation of excess tissue fluid. That would cause tissue fluid to go back in. Therefore, this is incorrect. And then for D, a decrease in blood pressure. Now, it's opposite to this point. Therefore, this is incorrect. Because if you decrease the blood pressure, the less tissue fluid is going to leave because there's a less hydrostatic force. Therefore, the answer must be B. Question number 32. Which statement is correct about a red blood cell that has just entered a capillary in respiring muscle? So, as we all know, a respiring muscle must always have an excess of carbon dioxide. So this means that Generally, carbon dioxide is increased in this area. Now, when carbon dioxide increases, it combines with water, and then by the action of the enzyme carbonic anhydrase, it forms carbonic acid. Now, this carbonic acid dissociates into hydrogen ions and bicarbonate ions. Bicarbonate ions leave the red blood cell. And then something called the chloride shift happens. Because chloride ions, which have a net negative charge, leave the red blood cell, to compensate for this, chloride ions go back into the red blood cell. Now let's see the suggestions that we have here. A. There's a net decrease in chloride ions. Now, this is not quite correct. If we would look for the single best answer, chloride ions would have a net decrease. This is correct. However, not for a red blood cell that has just entered the capillary. As we said, it is a long process. First of all, it starts by carbon dioxide combining with water, and then by the action of the enzyme carbonic anhydrase, it forms carbonic acid, and then when it dissociates and hydrogen carbonate leaves the red blood cell, and then a chloride ion enters back, this is when there's a net decrease in chloride ions. Therefore, this is correct. However, let's see if there are even better answers here, because this would be correct if it's said at the end. So if it had entered the capillary and then waited for a while. For B, there is a net decrease in hemoglobinic acid. Now this is incorrect. Hemoglobinic acid is formed when hydrogen ions combines with hemoglobin. This forms hemoglobinic acid. Hemoglobinic acid has a lower affinity to oxygen. This means that oxygen is released more easily. Now, this is incorrect. There isn't a net decrease. Actually, it increases because we said when carbonic acid dissociates, it releases hydrogen ions. And then when hydrogen ions combine with hemoglobin, this reduces the affinity, and then oxygen is released. Therefore, this must be a net increase in hemoglobinic acid, not decrease. Therefore, this is incorrect. C. There's a net increase in carbonic anhydrase. Okay, carbonic anhydrase is an enzyme. We know that enzymes are biological catalysts. They do not change in concentration. Therefore, this is incorrect. D. There's a net increase in carbaminohemoglobin. This is correct because, as you said, this is the first process. When carbon dioxide combines with hemoglobin, this is called carbaminohemoglobin. Therefore, this is an even better answer than the first one. Because, as I said, for chloride ions to happen, a net decrease in general in chloride ions, it must take a while. However, carbaminohemoglobin instantly happens when carbon dioxide, which is a lot in this case, combines with hemoglobin. Therefore, this is correct, and the answer is going to be D. Question number 33. The graph shows some blood pressure changes that occur in a human during one cardiac cycle. When does systole start and end in the right atrium and the right ventricle? So overall, how to interpret a graph like this is perfectly labeled for us, but let's show you a way of how to interpret it, just in case it shows up again. So here we have a mnemonic called COCO. This is for close, open, close, open. Now, we know at the bottom, generally, this should be the ventricular pressure, if we were looking at the left side. And above this pressure change is the pressure change in the aorta. So this is the ventricular pressure, this dashed line. And this smooth line is the pressure change in the aorta. Now, here we are talking at the bottom, we always talk about the atrioventricular valves. And above here, we are talking about semi-lunar valves. Now, I'll show you a way of how to actually figure this out instead of memorizing it. So, let's think about it. When the atrioventricular valves close and the semi-lunar valves open, what do you think happens in the heart in this case? It's going to be ventricular systole. Because as we said, when the pressure increases in the ventricle, what happens is that it pushes the semilunar valve open. And because the pressure in the atrium is now lower, this causes the shutting of the atrioventricular valve. Therefore, we can confidently say that atrial systole must start from this area here. So atrial systole must start AV, so start when the atrioventricular valves close. And then, for here, what closes? The semilunar valve. So we know when the semilunar valve closes, this means that now the pressure in the ventricle is not enough to push the blood out. This means that now it is ventricular diastole, and this is going to be the end of ventricular systole. So X is going to be the end of ventricular systole because the semilunar valve now closes and blood is no longer leaving the ventricle. This means that the pressure in the ventricle decreased, and now this is ventricular diastole. Now, regarding the right atrium, we know the pressure at the bottom is the pressure in the left atrium, not in the right atrium. And because we know that in the atrium it generally generates a lower pressure than the ventricle, because the atrium only needs to push blood a very small distance from the atrium to the ventricle. Therefore, this is going to be atrial systole. And the answer is going to be A. Question number 34. Which statements about all bronchioles are correct? So, a rule of thumb, you must know that bronchioles are different from the trachea by two things. That bronchioles don't have goblet cells and don't have cartilage. So this is how bronchioles are different from the trachea and the bronchi. They do not have these both. So neither goblet cells nor cartilage. So let's see the suggestions that we have here. One, they have goblet cells. We know that instantly this is incorrect. So we can work with this question by mainly exclusion. Two, they have epithelial cells. Yes, they must have epithelial cells. Three, they have muscle tissue. Yes, because we said it only does not have these two, goblet and cartilage. Therefore, the answer is going to be D. Question number 35. What is shown in electron micrograph? So, if we were to zoom in a little bit, we would see a biconcave shape. This biconcave shape is instantly going to look something like a red blood cell. Therefore, this must be a red blood cell. Now, we either have a scanning electron micrograph or a transmission electron micrograph. So here is a section from our free Topic 1 notes. Please find a link in the description box down below, which you can access our website and download these notes for yourself. Now, our notes mention that the beam of electrons is passed through a specimen before being viewed, and only transmitted electrons which hit the fluorescent screen can be seen. So this is for the transmission. Whilst the scanning scans the surface of structures and only reflected beams is observed. And then it allows us to see the 3D appearance. Therefore, here we could see a 3D appearance and we could clearly see the surface of the structure. We are not seeing a cut section as in the transmission electron micrograph. Therefore, this must be a scanning electron micrograph because we could see the reflected surface because of the reflected electron beams in a 3D shape. Therefore, the answer must be scanning electron micrograph and is going to be B. Question number 36. Which statement correctly describes infectious disease? So infectious disease is anything you can catch. Therefore, let's see the suggestions that we have here. They are diseases caused by environmental factors that are not passed from one person to another. Therefore, this is incorrect. It contrasts the idea of an infectious disease. B. They are diseases caused by a fault in the DNA that can be passed from parent to their offspring. This is incorrect. This is a mutation. Mutations are not contagious. C. There are diseases caused by lack of nutrition in the diet. This is incorrect. D. There are diseases caused by pathogens such as bacterium, fungus, protoxis, or virus. This is correct because they can be transmitted, and the answer is going to be D. Question number 37. Organism X produces molecule Y, which blocks the activity of 70 S ribosomes. So this instantly tells us that this is an antibiotic. since it blocks the activity of 70S ribosomes, which are found in prokaryotes. An experiment was carried out to investigate the effect of Y on bacterial cells. The diagram shows the results after a week. Which conclusions can be made from these results? Okay, first of all, molecule Y functions as an antibiotic. Yes, we mentioned this because when a concentrated solution of molecule Y was added here, so the cytoplasm of organism X, which is molecule Y, this means it could be used as an antibiotic because we could see here no bacterial growth around it. Therefore, it does function as an antibiotic. And because the question mentions that it blocks the 70S ribosomes, therefore this is correct. Now, the second option here, molecule Y cannot be released from cells of organism X. Alright, this is slightly tricky. We could here see the intact cells of organism X. So a normal living cell of organism X has no bacterial killing around it. So bacteria normally grow around it. Therefore, this means that in a normal living cell of organism X, the substance is not released. However, concentrated solution made from the cytoplasm of lots of organism X cells cause this antibiotic effect. This means that you have to actually kill the cell and take a concentrated solution of the actual substance in order to form an antibiotic. Therefore, it's not routinely released by organism X. You have to kill the actual organism in order to reach the molecules. Therefore, this is correct, and this is incorrect. And now finally, molecule Y would also affect the synthesis of proteins from nuclear DNA in a human cell. Now, this is instantly incorrect. The reason for this is that we know that nuclear DNA, synthesis of proteins from nuclear DNA forms mRNA. mRNA is translated in 80S ribosomes, not 70S ribosomes. Therefore, it does not directly affect the synthesis of proteins from nuclear DNA in human cells. However, it could indirectly affect if this would affect the 70S ribosomes of the mitochondria, then it might affect the process of translation. However, this is not likely. Therefore, the most correct answer is going to be incorrect, and the answer is going to be B. Question number 38. A patient had repeated infections from the same pathogen over a few months. The patient had been treated with the same antibiotic on each occasion. This might have caused antibiotic resistance. Which treatment could prevent an increase in the antibiotic resistance of the pathogen? So, an antibiotic resistance is when there has been some sort of a mutation in the actual organism, enabling it to overcome the destructive action of the antibiotic. Now, let's see the suggestions that we have here. A, a narrow-spectrum antibiotic to treat non-cellular pathogen. Now, this is completely incorrect. The reason for this is that we know that bacteria are prokaryotes. They are cellular pathogens. Antibiotics are not used for viruses, and viruses are non-cellular pathogens. Therefore, antibiotic is only used for bacteria, which are cellular pathogens. Therefore, this is incorrect. B, a repeat prescription for the same antibiotic at a higher dose. Now, the issue is antibiotic resistance does not need a higher dose. This is incorrect. Let's understand the mechanism further given an example. Now, for example, some types of antibiotic resistance happen when there's a mutation in some of the channels of the bacteria. So, for example, protein pumps. There has been some sort of a mutation that now prevents the antibiotic of doing its action on a specific protein pump because there has been an actual mutation. So even if you keep increasing the antibiotic at a higher dose, the mutation still exists. So even if you keep increasing the antibiotic, still cannot identify with the actual protein pump as an example. Therefore, this is incorrect. C, a specific antibiotic after testing for the pathogen. Yes, a specific antibiotic after testing for the pathogen, even if there's an antibiotic resistance, there might be an alternative antibiotic which is suitable. Therefore, this is correct. D, a wide-spectrum antibiotic to treat different strains. Now, this is incorrect because a wide-spectrum antibiotic might not be specific for the actual pathogen, and this would actually increase the antibiotic resistance. Therefore, C is the only correct answer. Question number 39. A patient with kidney failure can be given a kidney transplant. In a kidney transplant, a healthy kidney is taken from another person and put into the patient's body. The statements describe the next stages of the primary immune response. So the question mainly asks about the normal immune response, which order correctly explains how donor kidney cells are killed. So it's basically asking how the immune response responds to foreign bodies. In this case, it's considered the kidney cells a foreign body. Now, first of all, we know that we have T cells and B cells. T cells exist in the thymus and they are in the body. Now, T cells have receptors. These receptors recognize specific antigens. When a specific T cell identifies a specific antigen, now what happens? It differentiates into both T helper cells and T cytotoxic. But T helper cell is the first stage of differentiation because it releases cytokines, which indeed now activates the T cytotoxic. Now the T cytotoxic releases something called perforin, which from its name perforates foreign body cells. So it attacks the actual cells while the T helper cells releases cytokines. So that when the T helper cell, which is the first step, releases cytokines, and now these cytokines activate the T cytotoxic, which is the second step, these cytokines also now activates B cells. And now these B cells, which is the third step, differentiate into plasma cells and more B cells and memory cells. Now these plasma cells releases antibodies, which then these all responds to the target, which is in this case the kidney. Now, let's see the suggestions that we have here. We broken down the steps. We mentioned that the first step is going to be the activated T helper cells, which releases cytokines, and then this activates the T cytotoxic to release perforins. Now, let's look here. Here we have activated T lymphocyte secretes cytokines. This is correct. This is the first step. And then the second step we're going to have is the T lymphocytes multiply by mitosis and differentiate into T killer cells. Yeah, this is correct. This is the second step. And now the third step is going to be the T killer cells binds to antigens on donor kidney cells. This is correct because now the T cells and the T cytotoxic releases something called perforin. This perforin binds directly to the kidney cells and causes them to be killed and causes them to be attacked by releasing the substance called perforin. Therefore, this is correct. And now finally, the T killer cells release toxins that destroy the kidney cells. Yes, this is after this step, after identifying the antigen, the T killer cells release toxins. And finally, the B lymphocytes differentiate into plasma cells and make antibodies. This is the last step. Therefore, the answer must be B. Question number 40. The diagram shows the simplified structure of an antibody. Which statement is correct? So first of all, X and U are variable regions. And these regions bind to antigens. V is called a hinge region, and this allows flexibility for the variable region to bind to the antigens. And finally, W is called the constant region. The constant region binds to phagocytes for the desired antigen here to be engulfed and recognized by specific phagocytes such as macrophages. Now, which statement is correct? U and X allow the antibody to bind to two different antigens. This is completely incorrect. Yes, X and U could bind to two separate antigens, however, they are two different antigens. What I mean is that they bind to the same shape of an antigen, for example, rectangle and rectangle, however, two of them. But here, it means it binds to two different antigens, for example, a square and a triangle. This is incorrect. As you can see here, they bind to the same shape of an antigen, but two of them. Therefore, this is incorrect. Really bad wording here. B. V allows the antibody to be flexible to bind to two antigens. Yes, this is correct. It allows, this is the hinge region, and it allows, for example, the antibody to expand further and be more flexible, so it can bind two antigens of the same shape. So this is correct. C. W can bind to one specific antigen. This is incorrect. W is the constant region, which binds to phagocytes. They do not bind to antigens. Therefore, this is incorrect. D. X can bind with a specific phagocyte receptor. This is the variable region. They bind to antigens, not phagocytes. What binds to phagocytes, as I said, is the constant region. Therefore, this is incorrect. And the answer is going to be B. If you find this video helpful, please don't forget to subscribe to this channel and like this video. And if you have any further recommendation, please leave them in the comments down below.